I think if we were to make a brain on a chip iPS cell, we'd use your neurons on. All right. So it was a wonderful lead-in. I always use this analogy of Reese's peanut butter cups and chocolate and peanut butter for stem cells and microphysiological systems, because they really do need each other. And so with that as sort of a, I guess I'm hungry lead-in. The field of biology really was turned upside down when Yamanaka and also one of our co-founders, Jamie Thompson, showed that if you reintroduce transcription factors that control embryological phenotype, that you can reprogram a terminally differentiated cell and turn that back into a state that's essentially equivalent to an embryonic stem cell. I mean, there are certainly ways to slice and dice and look at them differently, but by and large, their ability to proliferate, to turn into all the different tissues of the body are shared between iPS cells and ES cells. And that's really, especially once the field was able to look at making human iPS cells and then ultimately differentiate them into tissues and be able to show that power of pluripotency can then be cascaded down through the embryological sort of milieu into different cell types and be able to use those cell types for basic research. It really, we're talking a very short order from 2006, 2007 in mouse and human to Nobel Prize 2 years ago to commercially available cell types of many different types are being readily used and created in different labs. And so it really has changed the basic thought process around cell culture and how this model can be used. And certainly one of the, as I mentioned, a wonderful complementary interaction between man on a chip or tissue on a chip type programs where you can bring in this concept of anybody's cell type. Anybody has a disease background or has an adverse event or has a unique property that you can then take that person's sample and blood. You need a small amount of blood. We can do it from about, not counting infectious diseases and safety nets, about 8 mils of blood today, and I completely believe that will go lower in the future. So really from a small amount of blood, you can reprogram it to an iPS cell, make that iPS cell into any different tissue, study that person and understand why something bad happened, something good happened, or predict whether something good or bad will happen. That's really the future. And I'm not talking about it today, but really you can see that this power will ultimately disrupt the way we treat patients. And you'll be able to, either through a HLA banked model of identifying an individual for transplant or taking your own iPS line and using that as a starting material for regenerative medicine therapeutic. This will be upon us before we know it. There are actually a number of programs up and running around the world. So I think as a bit of maybe pretext, it's useful to talk about why, from my perspective, and I really learned this through a lot of work in cardiomyocytes, but in other tissues it's shown as well, that there's a value for iPS-derived stem cell tissues over primary culture. And so sort of from a historical perspective, you think about, it's been a long time since we've been able to culture a cell. So Harrison showed with frog neurons floating in lymph that he could keep them alive. This is over 100 years ago. And then Guy showed with making a cell line from Henrietta Lacks of making a HeLa cell 60 years ago, but really a number of improvements, both in terms of culture, in terms of the conditions, understanding different cell types, and being able to take very obscure cell types and put them into culture. We've been able to do that. But the fundamental challenge has always remained the same, is that there's a compromise that's been made to do that. And you lose function, you lose phenotype, and depending on the cell type, it becomes something different. So, and you think about primary cells, you're limited at some level to access to them. So for humans, you need to isolate them. There's a timing question. You need unique access, and then again, this propensity for most cell types in culture, because of what you need to keep them alive, also compromises what their basic id is in vitro. And so really, this is what led not only the surge in microphysiological systems and different culturing techniques and better media and et cetera, but also from my perspective, why stem cells are a dramatic improvement over that, because you see things that you don't see with primary cell cultures in a number of monolayer applications in it. And as I'll show you at the end, a number of different models show an improvement over primary cultures, or at least show value. One of the main drivers why stem cell derived tissues, I think, have a leg up is you can make them from anybody, as I touched on. So this question of being able to understand a problem either retrospectively or prospectively is invaluable. And we can do these reprogramming, as I mentioned, in a small amount of material. You can do that in a footprint-free way, so you're no longer introducing genetic changes into that individual. You're basically using an episomal reprogramming method that after a period of passages is no longer there, so you have this sort of genetically identical iPS line from a small amount of tissue. And again, the key driver, I think, of iPS cell utility is this concept of being able to do this in anyone at any time. Yet, you also think about another aspect is genetic engineering. So you have this replicating line, you can then introduce any number of genetically engineered tweaks. So if you're interested in a point mutation for long QT syndrome, for example, you can correct that and have an isogenetic control for that individual. You can look at lineage-specific markers. So if you want to trace the production of a cell type and look at efficiency, if you want to ultimately understand safety and where that cell goes in a living organism, you can tag it so you can identify it. You can introduce factors that will drive differentiation towards a novel phenotype that can't be done with sort of basic culturing techniques. So it allows you to control the environment to the level where you can do what you want to do better, basically. And there are all sorts of new techniques. And CRISPR, as that technology becomes, and there's a number of good papers recently on making that more selective and targeted, that will dramatically open this up versus having to do an engineered protein like a TALENs or a zinc finger. You'll be able to do this with a mRNA, essentially, which will reduce the cost and drive that faster, which is being shown already in the field. As I mentioned, primary cultures tend to lose their phenotype in vitro, so anybody who's done work in primary hepatocytes has known that you have this sort of limited window of looking at least from a metabolic perspective, the utility begins to slide away. Primary cardiomyocytes is even much more dramatic. To keep them alive over a period of time, you need to introduce a fairly high level of serum, and what that does is causes the cells to stop beating and to start proliferating. And so from a cardiac biology perspective, that's not at all what an intact cardiomyocyte does. It basically has very limited proliferation in vivo, at least in a normal situation. So this really, again, is one of the main drivers of 3D culture, or microphysiological systems, is to prop up the system, is to either slow that curve or tip it the other way. But what we've seen, and we had the fortune of doing a lot of the early work in cardiomyocytes when I was at Hoffman LaRoche, and what we saw is that we're able to demonstrate that if you take an iPS-derived cardiomyocyte and just allow it to incubate, you can see a maturation. So if you look at gene expression, you begin to see a steady accumulation of age-dependent genes. So genes you'd expect to see as an adult are less expressed early on, and as you let them sit, even though they're cardiomyocytes and they're differentiated, they have more fetal phenotype, but over time they acquire a more adult one. And this has been shown by others as well, who had more patients, let them sit for months, almost a year, and they showed that you can acquire significant hERG channel activity, et cetera. So basically shows that you can, if you're patient, you can allow this to happen on its own. But we also looked at other methods, small molecules, and I'll show you a lot of data on microphysiological systems. Cardiomyocytes, if you stimulate them, they'll mature. So there are lots of ways, from my perspective, to make time go faster, or disconnect the phenotype from time and allow you to reach whatever purpose it is. And I'll show you some interesting data on monolayer non-aged cardiomyocytes work very well for predicting QT. But if you're looking at age-related diseases, or if you want to understand different functions that aren't yet there are certainly paths, including microphysiological systems, that will drive the phenotype towards a more mature state versus, waiting 18 years for it to mature. Sure.
You talk about the advantage over the normal primary cells in terms of losing their phenotype. Is the advantage of the stem cell that when they differentiate, they don't do that, or because you have stem cells there, you're constantly regenerating that particular population?
So,
could you repeat the question? For
sure.
The
So the question is it a recapitulation of the niche where you have stem cells that repopulate in a model versus they have a different phenotype in vitro? And it's more the latter. So what we've seen is cardiomyocytes is a good example. So the experiment that we published basically shows once you make a cardiomyocyte through directed differentiation, it's around day 20 in culture. If you let that day 20 cell sit, and we have data on day 20, day 28, 120, you see a progressive accumulation of age-specific cardiac markers. So they're maturing in vitro, and the others have shown this as well. The cells are purified population of cardiomyocytes in our hands, so there's no regenerative, progenitor cell in that mix. It's a 99% pure cardiomyocyte population that's not differentiating or not proliferating any further. So, for the most part, what it is a maturation.
No, I'm sorry. My question was, you said when you use primary cultured cells, they de-differentiate, lose their phenotype.
Yeah.
You said that does not happen when you differentiate the iPS cells into cardiomyocytes.
Right.
And so then they just hold their differentiation characteristics for a much longer period of time than the primary cells.
Yeah, it's more than hold. They actually continue to mature. At least in monolayer culture, cardiomyocytes have shown it. We've shown it with hepatocytes. Yes. So with that as a pretext, and really only as a means to show you what needs to be done to do this in an effective way, or at least one way, is I'll give you a few commercial slides on our company, because I think it gives perspective on what cellular dynamics has done to try to ensure robustness and stability to a biological product. So CDI is based in Madison, Wisconsin. We're the largest producer of iPS cells and derived products. We have a site in Nevada, and I'll talk about why that is in a minute. We went public about a year ago this month. We have 150 employees. Our company, because it was co-founded by Jamie Thompson, who's sort of the forefather of embryonic stem cell research and iPS cell research, we have a number of people who've come from his lab as well as other labs that have deep stem cell expertise, not only in iPS cells, but also in differentiating them into different tissues. We have a fairly large patent estate, and this is really to enable partners who use our cells, and me as a former, actually the first customer for CDI, needing to do research from a pharmaceutical company perspective, for example, so that they have freedom to operate. So you have to make sure that customers can do what they want to do in terms of study a drug, understand it, market it, and have the freedom to do that. It requires a large patent estate. So we're working with many different pharma companies and biotech companies, and we're beginning to, because of sort of the robust production process, beginning to think about how this manufacturing machine can be used towards clinical therapeutics as well. Core competencies range from making iPS cells, gene editing of iPS cells, differentiating them into different tissue types, and making lots of different tissue types, which I'll show you. So we have a panel of a number of cells from all the 3 different major germ layers, and we're sort of continuing to add different cell types steadily. The way we look at this is that when a person uses our cells, we want to make sure that they're as successful as possible. So we put an extensive amount of effort into the back end. So we have every time a new device is made or a new version of a reagent is made, we work effectively with that partner to make sure that we create a protocol, there's a process, that we train people, we go to their labs, they come to ours, etc. Really work hard to make sure that if people are willing to take a risk on us, that we can ensure them to be as successful as possible. So, and one of the keys for us is that everything that we make is highly purified, so at least 95% pure of the target cell population, and they're cryopreserved, so they're manufactured and stored, and then they can be distributed globally to anyone who is interested in using them. This year we've really begun to extensively allow for people to submit cell types of interest. So if there are a sample of interest, to be reprogrammed into an iPS cell. So, for example, interested in patients with rapid diabetes. We did this project with a partner, and they sent a number of samples. We made iPS cells and made different tissues from it to be able to study those tissues of rapid onset diabetes and how that can be used as a model. So we have a sort of an engine to be able to take a sample and make an iPS line from it, turn it into a tissue. So we can make lots of different cells, a sort of scale-up aspect of quality, quantity, and purity. You know, this is just metrics of what number of cells can be made per day. At the same time, we can start with any different iPS starting material. So we have a number of grants. This is one of the first ones was making iPS lines from 250 different patients that had the most informed patients from a genome-wide association study. And so being able to link SNPs from a patient population to functional output in a cardiomyocyte, that's really the driver of that collaboration with Medical College of Wisconsin. And this is really sort of one of the major activities for us, and this is why we have this site in California, is we were given a grant in partnership with Coriell to make 3,000 different, take 3,000 different donor samples and turn them into iPS lines from a number of different diseases. And all of these lines will be made sort of in this footprint-free method, will be banked by Coriell, and will be accessible to anyone who wants to access those lines. So this is really one of the first banking efforts that is actually up and running. So we're making lines already. And this we see as a wonderful catalog and an example of being able to create access to iPS lines of various different phenotypes. So really coming down to the last slide on the commercial side, and I'll get into some data. But to me, one of the things that really struck me and why we did the deal we did with CDI when I was at Roche was you really need to manufacture cells. And at the time, I had a lab with a couple of postdocs, and we were doing some work in trying to differentiate tissues. And really, we began to talk to them, and CDI spent $35 million on a quality management system to be able to manufacture things appropriately. So if my postdocs I had were biology folks. They weren't also supply chain experts. They weren't experts in GMP manufacturing and ISO manufacturing, all the things that are crucial to be able to do this so that when you make a cell today, it's the same as yesterday's and will be the same as tomorrow's. So those types of things are crucial. And really the field also improved a lot in terms of shifting media from culture-adapted media to controlled, defined components, shifting from feeder layers on mouse cells to being able to use recombinant proteins as the matrix, and really this control of reagents from start to finish, and then ultimately automation helped make this a much more doable thing when you then wrap it in a quality management system that can manufacture things to a spec. So I look at this as it's a necessary activity to provide access to everyone. So and we work, as I mentioned, we work with many different academic partners to take their methods and convert them to scalable manufacturing ones. So sort of for those who have pharma experience, it's sort of medicinal chemistry into process chemistry. And there's a wonderful example published on the microarray field. And I look at this as a very parallel process. So in the early days of microarrays, academic labs, pharma companies were spotting their own arrays and making their own microarrays, and it felt like that was going to be the way. And there was effort from a number of companies to begin to manufacture things to a spec. And that really, at the end of the day, became the driving approach. So people basically go to an Illumina or an Affymetrix today and access an array so that they can get back to the biology they're interested in and not worry about making their own spotting array facility. In the field for stem cell biology, granted, you're talking about a biological product and not a manufactured machine or entity, but it's very similar in that most people are interested in a biology or a disease process or a drug development process and not in how to make a specific neuron or specific cardiomyocyte. So I see that this, CDI is one of many companies that will be doing this, will be providing materials for people to get back to their basic area of interest. And this paper really goes through the history of how that happened in DNA microarrays. If you're interested, I can send that to you. So from my perspective, as I mentioned a little bit already, cardiomyocytes were the first cell type that went through this process and became manufactured to a spec. And so this data here shows the gene expression data that from cardiomyocytes, they look very similar to an intact heart. To me, this was the moment where I was like, wow, this is crucial. So this is the one with the little Roche logo there. You can see a very strong correlation. That dotted black line is a correlation to the expression level seen in vivo of the 500 most cardiac specific genes. And the correlation was 0.6, 0.7, so fairly high for an in vitro model to map to an intact tissue. And you compare that to cardiomyocytes in the inset, which is 0.1, so there's really no correlation. So this to me was the aha moment. And of course we went on and looked at lots of other things. All the major ion channels are there. All of the drug responses were there that you'd expect to see from a cardiac cell being treated with a drug that has a known phenotypic event. And really, this, to me, it was like we shifted the lab at that point from working on cardiomyocytes from primary culture to let's figure out how we can work with this company to manufacture these, because we can solve a lot more pharmaceutical problems per unit time than we ever will with primary cell culture. And one of the things, I don't know how I can do this. It's not a touchscreen, is it? Oh, it is. Okay, thank you. All right, so this is just one of the things we showed. And this is with using impedance, but we also looked at it with multi-electrode array. So impedance is basically just a low field detector at the bottom of a dish and allows you to see if that cell is moving. And as impedance, it's basically just looking at the physical movement of the cell. And what you can see, this is cardiomyocytes in a monolayer beating nice and rhythmically. And you add a drug, E4031, which was a class 3 antiarrhythmic that was abandoned from development because it caused exquisite arrhythmia. And you can see that the normal rhythmic beating is then discontinuous when you put in a drug E4031. This is 30 nanomolar. This is a relevant therapeutic concentration. And what you see on the lower left half is the tracings that you will receive from an impedance output. Nice rhythmic beating on the top line, and then as you step up in concentrations, you can see these partial incomplete contractions, essentially EAD type events. And what we went and did is quantitated what concentration would cause a greater than 20% of your beats to be arrhythmic. And we used that as a threshold then to determine whether or not that would be predictive of a drug to cause arrhythmia in vivo. So this IB20. And what we showed in this paper published in 2011 in Toxi, a number of well-studied molecules that induce arrhythmia in vivo will then induce arrhythmia at very low concentrations in vitro. So the IB20s are shown here relative to their ability to inhibit the commonly studied hERG channel, as well as induce QT in vivo and induce arrhythmia. A nice separation of drugs that would induce arrhythmia versus not. And obviously things like awabine and aconitine that go through more of a calcium-mediated inhibition mechanism won't inhibit the hERG channel, obviously, but cause arrhythmia in vivo. You can see that they will induce arrhythmia in these cells because the full complement of ion channels that are there leading to contraction are perturbed in this model. And again, similarly, molecules that will cause hERG inhibition but are, for various reasons, don't cause arrhythmia in vivo, you see a similar type of phenomenon. They don't induce arrhythmia in vitro. So this, from a pharmaceutical perspective, was a useful learning for us. We began to use this as a triangulation method for when we looked at hERG screening. Is that going to be a true risk? Is this worth continuing to pursue, or should we drop a molecule? It gave us context to what was otherwise a fairly error-prone screening system. And then we, of course, went on. This is a paper that's published beginning of this year, expanded the data set dramatically and changed the metrics that we used in terms of the mathematics of prediction. I won't go into it, but basically what we showed is that if you look at just beat rate, so normal beat rate, if that slows down, that's an exquisite predictor of QT prolongation. And it's around 90 to 95%, depends on which thresholds and which metrics, et cetera, that we sliced and diced in the paper. But so we then looked at the irregular squiggles, and if you use that as a prediction, you can predict arrhythmia in vivo about 80%. This was with as large of a data set as we could get our hands on from the sort of clinical QT world. And then nobody believes anything until their own internal drugs are tested on it. So we at Roche, we tested our own stuff on it, and it was about the same prediction, is that QT prolongation about 90-95% and arrhythmia prediction about 80%. So again, this gave us confidence that this is a useful model to predict drug-induced arrhythmia and be able to begin to test development candidates on it, which is the ultimate value assessment that a company can make. So if you think about this, really in very short order, moved from sort of a product idea to manufacturing it, to publishing it, to other folks were using it to test human safety and using those data as part of IND filings for showing that their molecule may not have a direct cardiotoxic effect. To ultimately, this is part of the efforts that HESI and FDA are going through to begin to reassess how they look at thorough QT trials. And ultimately, cardiomyocytes will be a part of this. There'll be an in silico piece. There'll be, it's comprehensive in vitro proarrhythmia, SIPA, which is pushing forward quickly. The goal is actually by next year to no longer accept clinical QT data, thorough QT studies, and then by the year after that, to change ICH 7B. And so these are not my goals. These are goals stated by Norman Stockbridge, who's the medical director of the cardiorenal division. And the HESI group is actively pushing to generate a lot of data to fill in that mandate or that request. But it's something where we know that in vitro and with an improved ion channel screening efforts, we can do a better job of predicting cardiovascular safety. That there's certainly a gap there, and there's been, not a lot of drugs have been pulled from the market for causing arrhythmia, but a lot of drugs have been stopped from development that would otherwise have a non-clinical, a mild degree of QT prolongation without a clinical sequela. And so this is really attempting to address that issue. Whether those dates will be sliding or not is to be found out in the next 12 months. So, but I give you that as sort of an example of even in monolayer, stem cell-derived cardiomyocytes are helping change the way we think about regulatory risk assessment. Our company, we have hundreds of customers, and so we've looked at lots of different disease applications, lots of different models have been tested and used, and I just sort of give you, a generic list of stuff that people have looked at. But really, one of the key drivers is these contrived or induced disease models. So things like taking our, taking our cortical midbrain neurons and treating them with A beta 42 and showing that you can mimic an Alzheimer's type event, and you can study that and actually learn which CDK proteins were involved. You can induce hypertrophy with endothelin. You can induce disease modeling in the liver. In the lower left is hepatitis. You can actually recapitulate the entire virus life cycle, which you can't do in primary cell culture, and allow you to take genotype 1 hep C patients, put their serum in a dish, and they'll infect those cells and they'll replicate. So it really is changing the fundamental tools that people can use from a drug discovery, drug disease process effort in monolayer. But when you begin to think about the next step, and this is really, for us, we're a manufacturer. We want to provide to whatever people want to use the cells for, and we're seeing a shift in application. So people are beginning both on the commercial side of making lots of different microphysiological systems, as well as from the granting side is applications of, the reason why we're all here is to talk about microphysiological systems. And so the way I look at it is, for those who play Super Mario, is you're taking taking something that works very well and powering it up. So you'll be able to look at better functionality, better maturation. You're able to look at complex multicellular interaction and ultimately suitability for clinical application, but the goal of having the same person's genetic material in multiple cell types and using that in a microphysiological system that better reflects the in vivo environment is really powerful interaction of the two. So clearly, having the native architecture similar, having ability to do deeper analysis, is why microphysiological systems are why they are growing. So in the last few slides, I want to talk a little bit about examples. And basically, these are a selection of just a few. But the sum total is that as we put stem cell-derived cells into microphysiological systems, what we're seeing is increased maturation, increased functionality, and improvement. So here's one example from Site 2 showing that if you take aligned, so it's a micropatterning effort, you align cardiac stem cell-derived cardiomyocytes, what you see is the sarcomeric structure improves, you see an alignment, force of contraction increases, and then interestingly, the resting membrane potential lowers. So that's an indication of maturation. Another set of work, this is from University of Toronto and Milika Radicic, showing that if you take hydrogel compaction of stem cell-derived cardiomyocytes with fibroblasts and put them around a wire that are on two posts, and you allow those cells then to adhere through that hydrogel, you can then look at post-deflection. So you can look at not only electrical properties of contraction, but you can also look at force of contraction, which is something that most monolayer systems are not able to assess. And again, you see the same type of things. So you put the cells into an environment that's similar to in vivo, they begin to, sarcomeric structures align, membrane potential goes down, and they begin to look more like an adult cell. This is from data from a collaborator, Jay Hickman, who's been doing micropatterning of our cells on a variety of different, both neurons, cardiomyocytes, and hepatocytes, and can show that, again, you see maturation, you see an improvement in response. He has a cantilever system that allows you to measure cardiac contraction and force again. And again, it shows you that alignment will improve the biology of these cells. These are data from Salman Khatani, who is a co-founder of Hepagen. He's back at Colorado State, but he's shown that when you put stem cell-derived hepatocytes into this model in a micropattern environment, you begin to see similar type of phenomenon with hepatocytes. You see maturation, you see CYP activity increases, and inducibility increases, albeit not to the level of primary hepatocytes, at least for the most robust ones, but to a level where I believe you can begin to do basic research on, not only to optimize that, but also to study whether or not a drug candidate or a molecule is metabolized through a specific P450 phenotype. And then lastly, this is sort of a different model. This is from Hemashir. Hemashir basically recreates human biology by mimicking the vascular flow. So they're academic experts who understand the modeling of blood flow in different tissues, and they can reproduce that with this sort of spinning disc. And in doing that, they show, again, maturation and an optimization of the phenotype with stem cell-derived tissues. Here we're showing data on liver, but we've got a number of different iPS cell-derived projects in process with them, again, showing maturation. So if you put a cell type into a model that improves their phenotype through microphysiological systems. So hopefully what I've shown you is that there's this power of iPS cells to improve them, improvement over primary cell culture, not only in terms of access, but genetic engineering, this maturing phenotype, improved functionality, but this ultimately, this ability to integrate human diversity is key. The aspect of manufacturing is essential to be able to do this appropriately. And, you know, ultimately, all of these things roll up to being able to ask better questions that people weren't able to ask before using primary cell culture. And I put this as a question mark, is now beginning to the point where we actually ask the question, are there regulatory decision-making applications that microphysiological systems coupled with stem cell-derived tissues can be used for in the near future? With that, I thank you for your time.
There was one quick question. Who's at the microphone?
For the cardiac data that you showed with the arrhythmias and so on, how many patients did that represent? Was that just one person, or? When you're looking at the arrhythmias and the effect of the drugs on
Yeah,
the,
they're, oh, you mean in terms of the stem cell-derived
yeah,
cardiomyocytes?
what kind of a population?
That is, I guess, the N is from a single iPS cell line, but the experiments have been done over and over again. We've made lines from patients with varying flavors of QT disorders, and you show that same phenotype. And we've had people ask, actually, where they'd like to screen on those, thinking that that would be a susceptible population. But the data we show is the IPS line is an N of 1, and we do it in multiple different wells. But it's a good question. It's actually ties into our work.
So our last speaker for this morning's session is from the US EPA, National Center for Environmental Assessment, Dr. Wei-Shui Chu, who is actually a rocket scientist in the past. So he's got 2 physics degrees from very well-respected universities, but he's a bona fide and card-carrying risk assessor. So we wanted to wrap this morning's session from the promise to more of a cold shower of a practicing regulator and trying to tell us how this will or will not make his job easier or more difficult. Wei-Shui.